So now what I want to do is talk about tests for between line divergence. So lots of scenarios are like this. Let's imagine you're looking in a fossil record and you've got a stratophenic sequence, which is basically you measure a series of populations of different strata. So over time you see the phenotypes drifting around. Is that drift? Is that selection? You do genomic data. You've got several species of Drosophila, and you look at a whole bunch of gene expressions. You see gene expressions either changing or not changing. Is that drift? Is that selection? How do you determine that? If I have a population, a laboratory population, and I sample it after, let's say, 50 generations of selection, I see a certain amount of divergence. Is that consistent with drift, too much or too little? The key about all these things is all we're doing is we're measuring traits. We have no molecular information. These are kind of old school tests. But you can apply them on genomic data. So even though they're old school, it doesn't mean you should ignore them because they're actually quite important. And about half this lecture will be applying these things to genomic data. So here's kind of the outline. Again, this will go over lunch. As I mentioned, I've got the first two lectures are rather long. The next two lectures today are rather short, so we should be easily done on time. I want to talk about how you quantify divergence. I want to talk about the short-term divergence. That's simply due to drift partitioning the variation that's there. I want to talk about the long-term divergence. This is due to mutation pumping in new mutations. So I've got Landy's Brownian motion model, which depends upon the effective population size. I've got the mutation drift equilibrium model that depends upon the mutational variance. I want to talk about something called an Ornstein-Uhlenbeck process. Basically, it's Brownian motion with a rubber band pulling you back to a value. It's a model of stabilizing selection with drift. Then I want to talk about time series-based tests, and then finally down here, apply all these to things like gene expression data. Okay? So now we're actually getting into the actual subject of the course. So, good place to stop and see if people have any questions. By this microphone is odd here because it makes me think my glasses have got a dirty spot. I keep on trying to do this, and no, it's just that microphone sitting there. Okay? No questions so far? Everyone's up to speed? And again, you've been very good, but don't be bashful about asking anything, right? Going to cover a lot of stuff. May have some machinery you haven't seen before, so want to encourage people not to be bashful. The overview. So I have a population. Here it is. And let's then imagine a mountain range comes between them. Or I take it and I found two laboratory populations. And I go back at time and look for the divergence. What can cause that? Well, drift can cause that, but selection can also cause that. Selection can retard the amount of divergence, stabilizing selection. It can accelerate the amount of divergence. So what's the between line variance from drift? Well, it could be due to partitioning. That is, if I'm initially a population with a big A, little a, eventually you get fixed for big A, you get fixed for little a, that will cause those populations to diverge. That initial variation, as it becomes lost over time because of differential fixation, by chance you'll fix different alleles, the populations will diverge over time. So the partitioning of the initial added variation, that's a short time scale divergence. Likewise, I can get accumulation of new mutations, and that gives you longer time scales. So both these things cause divergence. You know, as you might imagine, they use different tests. This one here depends upon the effective population size. This thing here typically depends upon the mutational variance. A number of tests are based on whether an observed amount of divergence is too fast. So I have to have some idea of what the expectation is going to be under drift. That requires me to have some idea about the effective population size or the mutational variance. Likewise, I could observe the divergence is too slow. I'm not seeing as much divergence as I want. Stabilizing selection can account for that. So how do we then determine what the scenario is? Is it too much, too little, about what we expect? It's like the, Goldilocks and the three bears, right? Let's first of all ask, if I have a population, here it is, I draw two founder populations from it, and I let them diverge by drift and I allow no mutation, what's the between population variance going to look like? So I measure the means in both populations. Over time, those populations are going to diverge, so I'll get a variance among those as those means diverge for two populations. For a bunch of populations, I measure that divergence by the variance between means. We can write the expression down. You basically get this. So this here, we've seen that before. That's loss of heterozygosity. That actually turns out the amount of inbreeding. So basically, the between line variance, that's just initial sampling. Let's not worry about that. And that then depends upon the initial variance. When I get fully inbred, that is, my population is fixed for all alleles, it's just twice the initial variation. So if I have a population, what happens over time is eventually, when they diverge and all the initial alleles are fixed, the amount of divergence between those two populations is twice the initial added variation. So the added variation is low, that divergence is going to be small. Several basic questions. Number one, I'm assuming that the environment is not an issue. Because obviously populations can diverge over time without any genetic change, because in population 1 and population 2, the environments have changed. We try to get around that where possible with laboratory experiments by growing things in a so-called common garden. I've got two divergent populations. I sample, I put them both in the same environment, I measure. I obviously can't do that in the fossil record. So there's an expectation that you're assuming that divergence you see is not environmental. That's a big expectation. Maybe you can take environmental measures to try to correct for that, but it's a big assumption. So then the thing is, if you assume that it's not environmental, how do you quantify the variation? Well, suppose instead of 2, I've got L replicate populations. So I have a founding population, and I draw 1, 2, 3, 4. The key assumption here is what's called the star phylogeny. When I draw each population, there's no covariance among them. So for example, I draw a population here, and later on I split out rows 1, 2, and 3. This population, there'd be covariance in here. So I'm assuming I don't have that. We can build that in. We're assuming we don't have them. We assume that I sample L populations once, and they're all sampled at the same time, so there's no covariance between any two of those, which would happen if, for example, I sample one population and then resampled it later on to make different populations. So in that case, a simple measure might be take the mean of the ith population, zi of time t, minus the overall mean, that's what the dot is. This is just a sample variance, right? That seems like a reasonable measure for the between line variance, which simply variance in means, is to compute from the means for each population and take that as my data and compute a sample variance on those values. If I have just 2, the square difference between them, you can basically show that's equal to d squared over 2. So if I take this expression and simply replace it with 2 populations, I can show that's equal to the square difference between the 2. So very often people work with 2 or many. In both cases, you can relate that to a between population variance. Now the problem is, this isn't a good estimator. And it's not a good estimator because the sample mean here is estimated with error. Here's the sample mean. It's the true mean. The variance in those true means is what we're trying to get at. And then there's an error. And that error depends upon the sample size. So if that sample size is small, that error can be rather large. So the way you get around this is the standard analysis of variance. So a one-way analysis of variance, I have several things I measure with replicate measurements. I can compute a mean squares between and a mean squares within. The mean squares within basically is the sum of the individual observations minus their specific means. The mean squared between is basically this expression here. And then I can adjust for that, and then I can get a clean estimate of that. So if we're looking at divergence, we want to look at the between mean variance, and the cleanest way to get that is to do an analysis of variance. Because it basically, if I simply use the means directly, I've got an error associated with their sampling. If n is large, it's not a big deal, but to be clean, you should always basically do that. If you only have 2, you can still use this expression. So estimate the between line variance with an ANOVA. That's your clean estimate. Yes? No, so remember, that gets taken in here. N is your, so what is this expression up here? What this basically says is, if I take my sample mean for population I, that's my true mean for population I, plus an error. So the true mean is the variance I want, that's variance between. The error in here, sigma squared z is simply the error of a single individual. So if I have n observations to get my mean, it's that. So usually the effect isn't much, but if you have small sample sizes, like you have a fossil record, and each population only has 10 individuals, it can be pretty big. So bottom line is always use ANOVA to get your between line variance. It's just cleaner. And the reviewers aren't going to hassle you. If you use a one-way ANOVA, you're not going to get hassled by the reviewers. If you use the difference between them, reviewers will come back and say, hey, even though the difference may be small, you still want to do it this way. Okay? So again, I think it's important as we dig down details to keep the big picture in mind. The big picture in mind is I've got some data. In this scenario, I've got a replicate population that I've sampled individuals from. And I want to ask how much have they diverged. And the most natural metric of divergence is, with more than 2 samples, is what's the variance in means when I sample those individuals, the between line variance. And you can express the difference between 2 means as a between line variance as well. What's the cleanest way to estimate a between line variance? Use analysis of variance, because if I simply take the mean estimates themselves, those are the true means plus error. I want the variance in the true means, and to get the variance in the true means, I use ANOVA to get that out. Otherwise, I get an error term that's built in. Okay? Just kind of nuts and bolts so far. So far, so good? So here is Russ Landy. Here's with Phil DeVries in Costa Rica. I actually have a picture many years later next to Phil, the exact same spot. So Russ was the one behind a lot of these tests. I like to throw pictures in here to put faces behind some of the names. In the 70s and 80s, much of modern evolutionary quantitative genetics was basically laid down by Russ. He did a lot of this work at the University of Chicago, so it's often called the Chicago School, along with Steve Arnold and Mike Wade, who we'll talk about a bit later on as well. So a lot of the tests we'll talk about here, Russ actually worked on when he was a grad student at Harvard under Dick Lewontin. And the name Landy you'll see on a ton of papers in evolutionary quantitative genetics. So there he is. So one of the first terms for tests for selection on phenotypes is Landy, who proposed a test for whether the between population variance was sufficiently different from that expected under drift. So again, all these tests are comparisons. So if your estimate of the affected population size is way off, your test can be way off as well. But we've already talked about if you have a certain effective population size. In a second, we'll see how you can write that in terms of an expected amount of divergence. In fact, basically here it is. There's an effective population size, there's the amount of divergence. We can then ask if that expected amount of divergence is different from our observed amount. If the observed amount is statistically significantly larger, probably directional selection. If it's statistically significantly smaller, probably stabilizing selection. Too much divergence, directional selection. Too little divergence, stabilizing selection. So how do we formally test that? Well, Russ computed a test statistic that was roughly F distributed. So it's time, I think, for a little refresher. Hey, I got it first time. Can people all see over here? Yeah, I know I'll try to move on the far edge. I hate these that basically have that. I could actually use the black backboard back there, but then I would be off camera. So let me just kind of remind you of a couple of basic things in statistics. Chi-square and F-tests. So I'll write xi, if I write it as this, it means that is normally distributed with a mean of 0 and a variance of 1. Okay? So that's a chi, that's an x. What's a chi-square with k degrees of freedom? That's just equal to the sum I equals 1 to k of xi squared. So the sum of k squared unit normal is a chi-square distribution. That's why you can add two chi-squares. Chi-square 3 plus a chi-square 5 is a chi-square 8. If I then take the expected value of a chi-square k is k. So if I then take chi-square k divided by k over chi-square L divided by L, that's an F distribution with k numerator and L denominator degrees of freedom. So the reasons that chi-squares come up quite a bit is expressions of the form sum of xi minus x bar squared. That's a chi-square. This is n. That's a chi-square with n minus 1 degree of freedom. Minus 1 because you've estimated the sample means. So a lot of things in statistics, you see chi-squares and F ratios because sums of squares where the x is normal. By the way, I don't need mean 0 or variance 1. If the variance is different, I've got a constant out front of here that takes care of that. Sums of squares for normal data are chi-square distributed, and ratios of sums of squares standardized by their expected values are F distributions. So a lot of things that come up in basic statistics are chi-square and F. This is our fixation on normal distributions. So questions about chi-square or F? Don't be bashful. You've probably seen it before. You probably have heard this before, but it's good to kind of get refreshed again. So what Russ basically did was he computed a test statistic that was roughly F distributed. F's kind of a funky thing. It's kind of this thing that goes between 0 and infinity. It's got a squished, like a big peak near 0, and a long tail that falls off. It's very asymmetric. So we look for either too little divergence or too much divergence. The tail really near 0 or the long tail. And basically, here's the idea. This is called this constant variance test, Landy 1977b. So what I observe, VBT, that's the observed between line divergence. For L populations, it's basically given by, given by this expression here. That turns out to be chi-square with L minus 1 degrees of freedom. So that's the observed between lines. Down here, that's the expected divergence. So basically, if I take that between line divergence and basically equate it to this expression and ignore this initial sampling, I get this. So that's the expected amount of divergence at time t, given the initial added variation and given my effective population size, and that's the actual observed amount of divergence. So obviously, a couple things here. I've got to have a good estimate of the initial amount of variation, added variation. I've got to get a good estimate of n sub e. N sub e is maybe less concerned than you're worried about, because you can basically find out what is the critical value above which drift is unlikely. And if that critical value is far below what you think the actual value is, then you're probably pretty safe. So in Landy's version here, we assume that this is known without error. So this is a chi-square normalized divided by 1. If you basically plug in L equals infinity, you get 1 down here. So the degrees of freedom are L minus 1 for the L replicate populations estimating that. And infinity could just make that without error. If you estimate this with some other procedure, let's say have 15 degrees of freedom, this would be L minus 1 and 15. Rush initially presented this without error, but you can easily modify that with error. And we talk about gene expression, and we'll come back and talk about this. The bottom line is I have a test statistic here. I can now look up values in tables. So let's go to a specific example. So Drosophila pseudoobscura, one of the many Drosophilas. I think there are, 2,000 species of Drosophila. So he had populations from Anderson in a 12-year divergence experiment, 5 populations, 2 of which were maintained at 25 degrees, 2 at 27, and 1 at 16. After 12 years, they were then all raised, the 5 replicate populations, in common environments, all at 16 and all at 25, and measured for what else? Wing length, right? The estimate of additive variation in these two populations were 0.88 and 0.77. The among population variation was 6.6 and 4.4. An upper bound on the number of generations was 150, and the affected population size always exceeded 1,000. Remember, larger population sizes means that the amount of divergence is less and less likely. So if you highball it, you'll be pretty good. So taking this lower estimate, if you just figure out what the F ratio is by plugging those things in, you get 50.5 and 38 here. And with 4 degrees of freedom, why 5 replicates, so L is 5, L minus 1 is 4. Both these were highly significant. So what you saw in this simple experiment was too much divergence relative to drift. Now, drift is given by T over N. As T over N gets, that quantity gets larger and larger and larger, drift becomes more and more important. So by taking if your effective population size was much smaller, then the drift might be more consistent with it. So one thing you can always do is take these data and go back and ask, how small does the effective population size have to be for this statistic to no longer be significant? You can work it backwards. And that's often how it's done, because it's another way of looking at things. So if you're worried maybe the effective population size is 2,000, the nice thing is you can try different effective population sizes. You can find the critical value above which drift is unlikely. That's an example of the so-called constant variance test of Landy. And that is a good place to break for lunch. So to frame where we are, I reminded you about these important concepts in quantitative genetics, this concept of an additive variance, which we often measure by the heritability, which basically gives us some indication about how a trait's ability to evolve. We talked about the additive variance. We talked about the effective population size and the fact that basically drift removes variation. One way to think about that is you can think about if I have a population here, split it in half, and the two populations start identical. As they undergo drift, the populations diverge because by chance they fix alternate alleles. Sometimes they'll fix the same allele, no divergence. Sometimes they'll fix alternate alleles. And so populations can diverge because the initial additive variation can be partitioned. They can also diverge because new variation arises by mutation, and that drives the populations apart as well. And so what we're now doing is talking about how we can formally do tests of selection when we're looking at phenotypes of single traits. And for example, trait measured at different points in time, or trait measured along a sequence. And we already talked about just to remind people, we talked basically about one way to measure divergence is to basically look at the between line variation. Then we talked about Landy's test, where you take your observed between line variation and you divide that by your estimate of the added variation times the time times the effective population size, and that gives you an F statistic. We gave a simple example of that. So this test here is called Landy's constant variance test. It assumes this variance stays the same, and basically where that expression comes from is this is how the initial variation of the population is partitioned over time due to alternate fixations. This is simply taking the initial variation and partitioning it. So for a short time scan, this is what happens because mutation hasn't really had much of an input. So that's kind of one of the early classic tests that Russ proposed for simply looking at how populations diverge on a short time scale. Let me introduce this and see if there are questions. So now we're going to look at longer term divergence tests. And the previous test assumes mutation's not really important. That means that T is much less than NE. Over long time scales, much of the divergence is generated by new mutation, and therefore we move from tests which are based upon effective population size to tests which are based upon the mutational heritability or the mutational variances we'll talk about in a second. So we're going to get into these in a second, but before I do that, let me ask if there are any questions or comments. And again, I want to remind people, we've been very good about this. Any topic is on the board. We have very diverse backgrounds. Likewise, I often speak very quickly. I get excited, speak even quicker. I'm actually ramping it down now. It's hard to believe. So if you need me to repeat something, I'm also happy to do that as well. Yes? Uh-huh. Yes, that's true. That's a great question. So what you basically need is you need an estimate of the initial variation, or you can take an estimate a little bit down the line and assume it's close enough to the initial variation. Because remember, this is basically how that initial variation is partitioned, right? So this is the variation you expect at a certain time, because basically t over n is a number that gets basically, that is the amount of, this basically 1 minus that is the amount of variation that's remaining. So that's the amount of variation that's diverged. So you need some sort of estimate on that. And again, the reason this has infinite degrees of freedom here is you assume that's known without error. If you have to estimate it, the estimate is the degrees of freedom you replace there as well. So it's typically not that widely used, but it's one that you see occasionally. But it's more typically done in these short-term experiments, right? We'll talk about longer-term things in a second. Yeah. Yeah, so the unbalancedness would basically come in terms of your ANOVA. So when you do the ANOVA to estimate between line variance, differences in the number of lines would get cleaned up, and the ANOVA would basically account for those differences in error estimates associated with that. Likewise, if you don't have a star phylogeny, so for example, suppose I've got 5 lines, and the idea is that all 5 lines were drawn from some initial line and haven't been connected since. You could have it where I draw 2 sets of lines, and then 10 generations later from each set draw 3 sets. That would give me a covariant structure. You can actually put that covariant structure into a mixed model to do the ANOVA that way and handle it that way. So differences in covariant structure among the replicates, differences in sample sizes, all that to estimate your between line variation you can clean up by putting in the right linear model. That's kind of how that's done. Okay? So here, and again, I'm not going to go through the details. You can read all about it in chapter 12. This basically is what the between lines variance looks like when you have mutation. And here's the critical part. That expression there basically tells you how the initial variation that's present decays. Because as t gets larger, this basically, this term here basically goes away. This term here is the new variation that comes in by mutation. You'll notice something here. The decay depends upon the effective population size here. Here, effective population size is no longer present. And one way to think about this is, go over here for a second. Let's see, I think it's this one, yeah. If I have, hang on, let me get something you can see. So if I have molecular data, for molecular data, the amount of heterozygosity is equal to 4 Ne mu over 1 plus 4 Ne mu. So the heterozygosity is set by the effective population size. If I ask how much the 2 lines diverge, if they're separated at time t, the divergence equals 2 mu times t. So effective population size comes in for levels of variation. For looking at levels of divergence, due to mutation, the effective population size does not enter. And we sort of see that here, and if I'm looking at how the between line variance increases over time. Remember, the between line variance is your surrogate measure for how the lines are diverging. It's a much more general measure than simply square difference between 2 lines, because it allows you to handle more complicated things. What you see here is that you have partitioning of the initial variation, then you basically have this constant divergence as a function of the mutation rate. So under and this by the way arises under the so-called incremental mutational model, the Brownian motion model, where my old allelic effect plus some random variation is my new allelic effect. Remember that was one of our models. Under that mutational model, divergence is linear as a function of the mutation rate and is independent of the effective population size. So if I have two large populations or two small populations, they diverge at the same rate. The large populations would have more added variation within them than small populations, but the divergence would be at the same rate because divergence simply depends upon the mutation rate. So here's kind of a curve that shows you that. This is scaled in terms of the effective population size. This is the fraction of the divergence at that time due to new mutations. Initially, none is. At basically, if you go down to, what is it, about 1.2 NE, you have about half the variation is due to new mutations, and about 4 NE, about all the variation is. So when you're down in this time zone here, T over N is tiny. That's the constant variance test. When you're in this time zone over here, where T is large relative to NE, that's a test that basically depends upon new mutation. And the reason is, tests down here, like Landy's test, basically ask about partitioning that initial variation. So you have some starting variation, that's it. Tests over here are concerned about the long-term cumulative effect of additional mutation that accumulates over time. So these depend upon basically NE. These tests over here often depend upon simply the mutational variance. So let's look at those. As I mentioned, under the incremental model, the between line variance increases without limits. Why? It increases as T times a constant. So for larger and larger T, it gets bigger and bigger and bigger. We talked about the house of cards or the regression model. And again, remember the house of cards model, it says if I have an allele and it mutates, the effect of that new mutant is independent of where it started from. Whereas under the incremental model, the effect of the new mutation is where it started from plus some random increment. The regression model, it's basically tau times the current value plus a random increment. Tau equals 0 gives me house of cards. Tau equals 1 gives me incremental model. And outside of the incremental model, you eventually reach this equilibrium level here. That value isn't important. The important point is you don't diverge without limits. You come to some limiting value. The flip side is it's pretty slow because you get there as a function of the mutation rate. So even though I mentioned these different models behave differently in that the incremental model basically allows you to blow up without any limits, whereas the other models, you approach some sort of limit. The approach to that limit is quite slow, so if the mutation rate is quite slow, it really doesn't matter between these because you're never going to really get this asymptotic value. You'll come a bit close to it. But the reason that's important is if your model comes to a limit, that's often taken as a symptom or a sign of stabilizing selection. That need not be the case. So one important point has to do, remember I gave you this table, actually it was in the last lecture. I gave you a table that basically showed that over a large variety of traits, your estimate of the mutational heritability, mutational variance divided by the environmental variance, that's about 10 to the minus 3, plus or minus 5 or 6 for a large number of traits. Those estimates are obtained by getting a very small population, ideally a single individual from an inbred line, then growing it up, and for a series of subsequent generations, looking at the parent-offspring regression. Initially at 0, and then gradually it builds up as you build up new mutation, new variation from mutation in your populations. So those estimates for the mutational variance are done in extremely small populations. The reason that's critical is we have this notion here of 4Ne times S. If this quantity here is bigger than 1, it means the time scale over which allele frequencies change by selection is much quicker than the time scale over which allele frequencies change under drift. In other words, selection overpowers drift. When this is less than 1, it means the per generation noise 1 over 2n in allele frequency is much bigger than the per generation noise 1 over s in allele frequency, therefore drift swamps it. So in a really small population, selection is very ineffective. And so you might have a lot of your alleles that arise are deleterious. In a small population, suppose they have a 10% deleterious mutation rate, or suppose S is 0.1. That's a huge selection disadvantage. But if your effective population size is 2, you're still less than 1 and drift overpowers those. So those estimates of the effective mutational variances in small populations probably don't translate well to large populations. In a small population, a mutation arises. It's got to be really, 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 really deleterious for it not to be recorded as contributing to the trait, because the small population, remember super small, drift overpowers. In a large population, deleterious mutations are probably removed fairly quickly and therefore don't contribute much to the mutational variance. So the bottom line here is, even though I've phrased it as the populations diverging independently of the population size, because the divergence is time times the mutational variance, the mutational variance probably itself gets smaller as population sizes get larger. Why? Because as population sizes gets larger, more of the new mutations, which were deleterious and not scored as deleterious under small populations, now become deleterious and don't contribute. So as NE increases, the fraction of mutations that are effectively neutral likely decreases. So you do probably get the fact that divergence actually slows down as NE gets larger. Why? Because the mutational variance, the part that can actually contribute, gets smaller. Think of mutational variance as being this big, and the things on the extreme are deleterious alleles. It's this big when you have a small population. As you remove those deleterious alleles, it becomes much smaller, and therefore the effective neutral mutational variance is probably much less than the values that are estimated in these small population sizes. Just something to be aware of.
That's not because of selection. Just by chance you have less deleterious.
No, that's a great point. So that's a very good point. Let me clarify that. Here is, let's say, here's the distribution of selection coefficients. It's just something over here, right? And that's 0, so this is S. So basically, as long as you have 1 over 4 NE, 1 over 4 NE, everything in here is neutral. It's selected against, but from a population genetic standpoint, drift is stronger than the strength of selection, and so effectively selection just is ignored. As my population size gets smaller, the range gets bigger. As my population size gets bigger, the range may be only this big. So the red may be the range that if I take the same distribution, the red is the range of things which are neutral in these experiments to measure the mutational variation, where the effective population size is small. If I go to an actual population, of this range here, the actual part that's effectively neutral, given the large populations, is tinier region. So if this whole thing is, let's say, sigma squared n, that may be only 1/20th of sigma squared n. As my effective population size gets bigger, that region gets smaller and smaller and smaller. Not due to sampling, it's just due to the fact that when you've got selection, as your population size gets smaller, selection for and against things become less efficient. So in large populations, selection is very efficient, and therefore things which are slightly deleterious and wouldn't matter at all in your accumulation lines now become quite deleterious and are removed from your population and don't contribute much. So the point is, estimates based upon using the values for the red, which is what we use, may be quite misleading when you go to larger populations. Just because, you're in here, your design is such those alleles behave as if they're neutral. Not because of anything other than the fact they're in a small population. The key point. Okay? So we call these rate space tests, and we can frame them a couple of ways. We can frame them looking at the expected equilibrium value, which depends upon any of the mutational variance. We can ask if the values of NE are too large or mutational variance are too small. So basically, small values of NE, lots of drift, lots of divergence. Large values of mutational variation, lots of divergence. So if I see divergence, how large does this have to be for it still to be consistent with drift? Because below that, you're not. Likewise for here. So the way we do this, and I'll show a picture in a second, is we find the largest value of NE that's consistent with drift. If my actual value is above that, I reject drift. Because remember, as my population size gets larger, drift takes longer to work. Or I could also express it in a, so I could have a critical effective population size. If my actual value is above that, I have too much divergence relative to drift. Or I could have a critical mutational variance. If my mutational variance is above that, it's consistent with drift. If it's below that, I reject drift. So here's the picture. So red implies a significant result. So remember, when effective population size is big, drift is small, divergence is small. When my mutational variance is large, you have lots of drift. So basically, the blue areas can give you lots of divergence consistent with drift, either small population or large mutational variance. So I have a critical value. If my effective population size is above that, I have too much divergence relative to drift. I have a critical mutational value. If my mutational value is below that, I've got too much divergence relative to drift. And tests are set up using either one of these. So stabilizing selection, it's the opposite, right? Stabilizing selection, you want a little change. So you have a population size. If you're below that population size, you're consistent with drift. If you're above it, you're not. It's the exact opposite of what we've seen before for divergence. And here's the picture. So if my population size is large, drift is slow. If my effective population size is small, I get a lot of divergence. And the thing is, we're now observing too little divergence. So what it basically means is my effective population size, basically, there's a critical value. If I'm below that, I have too much divergence relative to drift. Likewise for the mutational variance. If my mutational variance is above that, I have too little divergence relative to drift. So remember, if I have a large population, it slows down divergence. If I have a small mutational variance, it slows down divergence. So in that case, a large population size that's too large or a mutational variance is too small give me a signal of directional selection. And likewise, a effective population size that's too small or mutational variance that's too large relative to some standard we'll talk about in a second means I've got too much divergence relative to what I expect under drift. So that's the basic idea. So Landy, same Russ Landy, he had a longer divergence test, and he called these Brownian motion models. So the idea under Brownian motion model, and this looks very much like, in fact, this is exactly what we saw under constant variance. Here, I'm assuming the variance doesn't change. Now, Landy's initial concern was the fossil record. How do you measure heritability in the fossil record? Well, maybe you can find parents and offspring dead next to each other, but really unlikely. So one way Landy thought was, well, you know, for a lot of these traits, heritability is pretty well known. Morphological traits. And further, heritability is in a relatively narrow range. So we can actually go in the fossil record and we can actually look at the phenotypic variance in the fossil record. So heritability times phenotypic variance is added variance. So Landy basically expressed these tests in terms of take a known heritability, and you could try different values, and use that by the observed phenotypic variance to get that. So the key here is we assume this heritability is constant over the sequence. If I have a finite population size and no mutation, it's going to be declining over that sequence. This is why mutation comes in. Conversely, my undergraduate advisor, Michael Torelli, and John Gillespie noted that, wait a minute now, if you have drift occurring and mutation, you're at mutation drift equilibrium, and now your added variance is given by this. So if you substitute that value for additive variance into here, you end up getting our result here. Divergence is simply time times the mutational variance. So this is now independent of the effective population size because this depends upon effective population size, and that does, and they cancel. So you have these two different versions. This is called the mutation drift equilibrium version. This is called the constant variant version. Which one you use is potentially problematic, as we'll see in a second. Now, a reason to maybe argue against this one is that value is too high. And what you can basically show is for known values of mutational variation, once your effective population size gets above 1,000, your heritability is basically 1. It doesn't happen. And the reason what's probably happening is as your effective population size gets larger, sigma squared mu starts to decline. Again, for this argument over here. As you pick up, as more and more alleles now become deleterious because your effective population size is larger. So the fact that this is independent of effective population size makes the assumption that this thing here doesn't change. It probably does change. So because of that, I think this is probably a cleaner version to use. Well, let's go through a couple of those. So you can basically, under the Brownian motion model, basically figure out what the critical values are. And Landy had basically a one-tailed test, too much absolute divergent. You can then plug it in and show the critical value is given by 1.96. So the bottom line here is what's the lower bound on the effective population size, above which there's too much divergence to account for drift. Well, it's simply equal to times the heritability times the time divided by the amount of divergence you observe. So bottom line here is if I know time and heritability, under this model I get a critical value for the effective population size. If I'm below that critical value, I can have all the divergence I want. It's consistent. If I'm above that critical value, there's too much divergence relative to the amount of divergence I'd expect. Remember, large N equals slower divergence. It turns out that this value here, if you think about it, is probably a little bit off. This is a one-tailed value. And so what Landy was assuming, this is an argument that Tyranny made in that paper, Landy was assuming you had too much divergence. Since I take the absolute value of D, too much divergence basically means that these tails here together are alpha over 2 and alpha over 2. But Tyranny also pointed out that a more reasonable test is the divergence abnormal relative to drift? That could mean too large, or it could also mean too small. In here, where these areas are alpha over 4, so if you sum these 4 areas, you get alpha. So a 5% critical level would be a value then that gives z of 5% divided by 4 here, 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 and here. So the original paper basically only considered these two tables. If you put in these, you get lower critical values. You basically, the critical values then become about 5. So you go from about 3.8 to about 5 if you consider a two-tailed test that is jointly too much divergence or too little divergence. This is only testing for too much divergence. Tyranny argued that's not the correct hypothesis. So here is the critical value for too much divergence, again at the 5% level. And likewise, here's the critical value for too little divergence relative to drift. So there are details in there, right? Those details you look up, those numbers you look up, either from the slides or from the book. Let's think about the key idea. The key idea goes back to these figures here. Whoops. If I have a test that's based on the effective population size, as my population size gets larger, drift becomes more unlikely. So I have a critical value. If my effective population size, I think, is bigger than that critical value, drift is unlikely. I could also take the same test and instead of basing it on effective population size, base it on my mutational variance. If my mutational variance is below a critical value, I have, again, too much divergence with respect to that. Remember, large N means slow divergence, and likewise, small mutational variation means slow divergence. So I get critical values for testing for excess divergence, and you flip those around to critical values for too much divergence relative to drift. So I think an example puts these together. These aren't the 4Ms that Reinhardt looked at, but gives you some idea about foraminifera, right? These are little tiny protists that make these calcium carbonate shells that fossilize really well. So there's a lot of work done with them because there's very good fossil records. They've got lots of morphological features. So what the investigator did here was he noticed a change of about 1.5 standard deviations in about 500,000 generations of, I think it was a test size, a body size characteristic. You take a typical heritability for a morphological trait of 0.3. Again, if that bothers you, you can try different values and see how the results change as you change heritability. You can then go ahead and plug in the numbers back in here. This is that one-sided test, a test only for excessive divergence. You then get a critical effective population size of about 250,000. So if your effective population size is below 250,000, that amount of divergence is consistent with drift. If your actual effective population size is above 250,000, that amount of divergence is too excessive. Since the census size was over 1 million, we mentioned all the problems of census size and effective size, but the bottom line is this seems to suggest you had too much divergence relative to what you'd expect under drift, because you expect the effective population size to be much larger. Again, there are the usual caveats. This is a fossil record sequence, so you're assuming that all the phenotypic change is due to genetics, not to some environmental trend. If you had some environmental trend, maybe warmer gives you larger bodies, you of course have a false signal, but you're basically assuming that's sort of not an issue. So if you use the two-sided test, so one-sided test simply means I'm asking if there's excessive divergence. Now, you're biased because you observe amount of divergence. That shouldn't bias you into testing for too much or too little. Your naive coming in should be, is the amount of divergence consistent with drift? That means, is it about right? It's not too big or not too small. So if you use that view, then this critical cutoff threshold here gets replaced by about a 5, and the effective population size, depending upon your estimates of the heritability, increases to about 340,000. Again, heritability has to get to be about 0.7 or bigger for that cutoff size to be within the range you think the actual population is. So using the version here based upon critical effective population size suggests that this amount of divergence is too much relative to drift. Now, conversely, you can take that same Landy test, and instead of dividing here by twice the added variation divided by NE times time, you can then plug in the equilibrium value. That's the expected amount of divergence you expect by mutation, another way of looking at it. And then you can basically do a similar test and put critical bounds on either mutational variation or the heritability. And when you go ahead and do that and rerun that same experiment, using that same data, you basically see that if the mutational heritability is more than this critical value, then drift is not rejected. Remember, a higher mutational heritability means more divergence. So you have a critical value. If you're below that, there's too much divergence. If you're above that, it's okay. So what is that critical value? It's that. And given a typical mutational heritability is 10 to the minus 3, this is several orders of magnitude above the actual value. So from the MDE version, this exact same data says divergence is not excessive relative to drift. So right away, we've taken the same data and gotten very strong conclusions. If I just use effective population size, I can clearly rule out drift, unless effective population size is really skewed relative to census. If I instead frame it in terms of mutational heritabilities, it's totally consistent. And the reason for this basically comes down, I think, to this idea here. The MDE test assumes that as you increase effective population size, sigma squared m does not change. And by the value sigma squared, I mean the part that evolution can use. If a lot of that is because you've got deleterious alleles, that's not usable. And so I think the reason you get the drift not being rejected in the mutation drift equilibrium is you're exactly seeing this. Your measure of effective population size affects the mutational heritability, the effective mutational heritability. And therefore, everything else being equal, a better thing to simply use is what's the observed thing we see. This is what we observe compared to the neutral expected value. So if you plug in this value, there's a test based upon effective population size, that's probably a little bit cleaner. But it does point out the problem that these tests can have, and that we get these dramatically different conclusions based upon what seems to be an equivalent assumption. Yeah. Well, you can't. And the answer is, suppose this is what you found when writing a paper. You'd obviously report them both. Then you come up with some clever bullshit scenario as to why one works for the other. I think the argument that makes sense is the mutation drift equilibrium value assumes the equilibrium variance goes to 2ne sigma squared mu, right? That effective population size times mutational variance. That gives you too large an additive variation. The heritability basically goes to 1 for, effective population sizes over, let's say, 10,000. And what that's not accounting for is that you have a certain amount of variation that's there from new mutations. But a lot of that is deleterious. You don't catch that in your small population estimators. As you go to larger populations, the amount that's there versus the amount that's usable are quite different. And it's the usable ones you want because those have got to be fixed for you to see this. So because of that, I tend to like the effective population size versions a little bit better because I think you can argue more logically where they're coming from because you have this problem. But again, be aware they're out there and be aware you can read them both in literature. So there's a large literature of a lot of the, if you go to like paleobiology, like in the 70s and 80s, there's a lot of discussion of tests like this. We don't. Some of the most interesting stuff comes out of Mark's lab, the Blows lab, where they basically shown that you can do indirect studies. We don't know these things directly because they're hard to get at. You could do a clean experiment. As far as I've known, no one's done it. And the clean experiment would take an inbred individual and basically estimate sigma squared m at different effective population sizes. Let's say 1 and 10 and 100. You should see those numbers go down. Hasn't been done. It's a lot of work to be done. It'd be hard to convince an agency to fund it, but it'd be an important thing to do. Selection is always out there. Remember, this is selection against mutations. So we think about selection. Selection is always occurring because most mutations that arise within your coding sequences are deleterious. They get kicked out. Either they're at very low frequencies and they get removed, or you don't see them at all. They never contribute to divergence. So here, I have new mutations that are arising. A mutation that influences your trait may have effect on other traits, and therefore may have fitness consequences, even if your trait isn't under selection. And therefore, we call it pleiotropy. When mutations arise, they probably affect a lot of things. And one of the unknown questions in evolution, I think someone mentioned it over here as an interesting topic, was just how pleiotropic is a random mutation. Furthermore, there's the issue of when you have a pleiotropic mutation, do you tend to have a constant total effect of mutation summed over all characters, or the same effect on all characters? That Euclidean superposition model versus the other model also makes a huge difference. See chapter 26, right? So there's a lot of stuff that I'm not going to talk about. But there's a lot of subtleties that underlie these that people are still in the process of working out. So I guess one of the big take-home messages is everybody loves to test for selection, right? Because nature's out there, it's shaping things. As a biologist, you look at stuff and you just know that features of your organism have been adapted and shaped by selection. You don't know which ones, They may not be the ones you're looking at, but you know they've happened. The problem is all these tests we'll talk about, trait-based, marker-informed trait-based test, marker test, direct measurements of traits in nature, all of them have significant biases. Most of the literature results are probably incorrect or not properly stated. That's the sad part about this. Maybe if that was a leading sentence in the course, no one would show up. But it's good to be aware of the limitations which are out there, so when you read the literature and you're working on something, you have a good framework to discuss things from. Other questions or comments so far? Yes? That's a great question. So basically, suppose I have a trait like height, okay? So obviously, as genes mutate in the genome, some of those genes that mutate have variants that influences height. So so each generation, new added variation in height will be generated at some small level by new mutations. So how do we measure that? Well, how do we measure added variation? We can measure it a couple ways. We can basically look at parent-offspring regressions, or we can actually look at the divergence between identical lines. So mutation accumulation experiments basically start with an inbred line, either brother-sister mating or a self line, and then basically ask what happens to brother-sister mating, what happens to parent-offspring regressions over time. That would basically means we're accumulating new additive variation. Or I can ask, how do they diverge over time? They'd only diverge if there's added variation there. Both those type of experiments are typically done under very, very small effective population sizes, 1 to 5, for reasons that make them easy, also to capture everything. Those kind of give you an upper bound on how big the mutational variance could be, an upper bound. Because what we want is not the mutational variance, we want the operative mutational variance. So operative simply means, okay, those mutations are occurring in nature. What fraction of those are neutral? Because a lot of the mutations that arise are deleterious. So we want to not count those, otherwise we'd overcount what the actual rate of effective input is. Those small population studies basically give you kind of an upper bound on how big that could be. As you add larger and larger populations, the fraction of those that are usable becomes smaller and smaller. Why? Because things that are under weak deleterious selection now become under strong deleterious selection in large populations. Not because S has changed, but because 4NES has changed. So the small population values are kind of estimating the upper bound. In real populations, you want to know what fraction of those are actually effectively neutral. And that fraction is going to get smaller and smaller as your population sizes get larger and larger. Sure, exactly. So for example, the estimates on height are, human height are on the order of, several thousand genes, right? If you look at life history traits, so the comment was made that the heritability is kind of an odd measure. Another measure to look at what fraction of variation there is take the variation divided by the mean. It's called mean standardization. It's called evolvability. When you do that, you see that life history traits have larger amounts of added variation when you mean standardize them versus morphological traits. When you variance standardize them, they have smaller. So things that have, and the explanation for that is for life history traits, a lot of genes can mess you up. Like viability. You can get killed or be unhealthy by a lot of different mutations. Whereas eye color may be 1 or 2 mutations. So certain traits, number 1, probably have more hit sites. Number 2, they probably have more of a variation when you do get a hit in terms of how much that changes the character value. So those are the things that go into basically accounting for what underlies the mutational variance. It's still one of the great unknowns. One of the things that's interesting is there's some inconsistencies between mutation rates and number of loci. In the sense, we know how big how much mutational variance contributes. We have a good idea about that in small populations. To get those values, we either need a much larger number of loci than we anticipated, tens of thousands, or much higher per site mutation rates, 10 to the minus 4. Nucleotide mutation rates, 10 to the minus 9. Look over entire gene, 10 to the minus 6. So some inconsistencies there we're still trying to fight our way through. And again, if you're interested, I keep on having to, but chapter 26 talks about that in like 70 pages. There's a lot of stuff there. Other questions or comments? So we have these rate-based tests where you either set a critical effective population size or a critical mutational variance or mutational heritability, sort of the same thing. Now, those are tests based upon the incremental model and Brownian motion. So basically, populations diverge by t times some amount over time. Another model is the so-called Ornstein-Uhlenbeck model from physics. And Ornstein-Uhlenbeck model basically says that the mean value, that is the mean change, if you're currently at x, equals a times some value minus x. So what this means, if x is at theta, there's no mean change. But as you move away from x, you get a larger restoring force. The idea is, think about Brownian motion, the particle bouncing around. And think about putting that particle on a rubber band stuck at 0. As the rubber band gets stretched out, you get more restoring force. As you're close, so the larger A is, the more the restoring force back. That's an Ornstein-Uhlenbeck process. Model from physics. It's a good model for stabilizing selection. Theta is your optimal value. As you move very far away, there's strong selection to push you back. If you're close, there's weak selection. So another model for divergence is the trait is under stabilizing selection. And you can go ahead and work out what the Ornstein-Uhlenbeck model is, and what you basically see is the mean at time t essentially becomes the optimal value. The variance, instead of going to infinity, approaches a constant. So the variance, instead of continuing the between line divergence, instead of continuing to get larger and larger, asymptotes out. And so if your data are under, your traits under stabilizing selection, what you expect over time is you expect an asymptote in your divergence. And this, for example, is what people see in some expression data. You don't see a linear divergence over time, you see an asymptotic divergence. The problem is it can also arise if your mutational model is different, if it's a house of cards model or a regression model as opposed to an incremental model. So, summary, again, we'll come back talking about Ornstein-Uhlenbeck in a little bit. Under Brownian motion model, the mutational increment, under neutrality, the means diverge linearly without limit with respect to each other. Under the house of cards or regression mutational model, eventually you reach a limit, although it may take quite a while. This time to scales as the mutation rate, not the mutational variance, the mutation rate. So a limiting amount of divergence could occur because I've got stabilizing selection, Ornstein-Uhlenbeck, or it could occur because of some feature of how new mutations arise. So people usually don't talk about this. When they see a divergence that stabilizes, usually think about stabilizing selection. This remains a formal possibility. That's the first type of tests based upon phenotypes diverging over time. In that category of tests, what we're basically doing is we're basically taking phenotypes and having some measure of effective population size or mutational variance or time. So I've got some external comparison to look at my look at my between line divergence. So look at VB, my mutational, sorry, jet lag is kicking in. Look at how the variance in between populations. I've got some standard to compare that to. The next category of tests are entirely from paleontology, and that's simply I'm giving you a time series. That's it. No external information like effective population size guess or time guess. Just that time series. Can you say something just based upon that time series? That's the next category we'll look at. Before I get there, though, we're getting, some of this stuff can get a little technical we've talked about. So again, take the overview. I call it the grandmother-grandfather test. Another way is there was a comedian years ago called Father Guido Sarducci. And see, our age is giving ourselves away. And he had something called the 5-minute university. And he said, everything you learn at university, I can teach you in 5 minutes. No, I can teach you 3 minutes because you have to have a minute for registration and a minute for graduation. And he said, 10 years after you took your economics class, what do you remember? Supply and demand. 10 years after your Spanish class, ¿Cómo está muy bien? And the bottom line is that there's a key idea you remember, and then you go back and look up the details. What's the key idea for the tests we've talked about so far? If you have data on amount of divergence, there are tests that allow you to ask if you see too little or too much divergence for an assumed heritability, effective population size, and time combination. The actual details on that you can look up. That's the key part to take away, right? Okay? That's the key part so far. A lot of information for that key part. But again, what you remember 5 years from now is that key part, then you go back and look up the details. You're not going to look up, is it alpha over 4 or alpha over 2? That's a detail, but knowing it's out there then makes it easier when you read about it again. It's not quite as foreign. So a question before we move on to time series-based tests. Yes? Yeah, that's a great question. So Ornstein Uhlenbeck is the model for how trait means change. House of cards and regression are models for basically what happens. Let's go back to those. So here's the approach. And the approach depends upon the mutation rate and time. It doesn't depend upon tau. So basically, they asymptotically should approach their values at about the same amount. And the key here, if mutation rate is small, 10 to the minus 8, then as long as you're looking at time scales under 1 million generations, it's not really an issue. So for long time scales, distinguishing an asymptote due to Ornstein-Uhlenbeck, making sure you're not too far away, or due to the fact that you don't generate new variation without limits, you can't distinguish those two. And that's the key part about this. Not that there is any way to do that, but simply be aware you have these limitations out there. Okay. I actually like these categories of tests because I think they're nice. So the above tests we've talked about are based upon external comparisons. One has 2 time points or a set of populations that diverged a given amount of time ago, and a guess for either T, NE, mutational variance, or VM. So I basically am comparing my data against some external reference standard that I either feel good about or I try different values and ask how uneasy do I feel about the values I need to account for drift. A second type of data is what's often called the stratophenetic, so stratigraphy phenotypes series, a time series of means for a population over some part of a fossil record. There are lots of these things. These are based upon internal comparisons, so time series tests basically just take this data and nothing else. Here's the data, bing. That's the data you have. Hopefully your data is a bit more in focus than that. So if you just see that, can you use that, just that graph by itself, to ask if that amount of divergence is excessive or not excessive? Is that consistent with a random walk? The answer is there are several tests that allow you to do that. So they're nice in that they just rely on the internal things and it's typical data you get. So Dave Raup at the University of Chicago, who passed away a couple of years ago, was kind of the founder of these things. Here he is holding up a nice ammonite. And Dave was one of the people that got statistical methods into paleobiology. A lot of the debate for analyzing these time sequences comes from Eldridge and Gould. Gould also passed away as well. And Eldridge and Gould basically argued that the fossil record has a certain pattern. And what Raup was basically asking was, what statistically can you say about the confidence of that pattern? In particular, around the 1980s, an idea called punctuated equilibrium, which his detractors called evolution by jerks. And the idea is, in the fossil record, they claim you have large amounts of stasis and then rapid shifts. You have an equilibrium that's punctuated. So that would be quite striking. And what they argued was that this pattern suggested typical Darwinian natural selection wouldn't be sufficient. You probably have selection on the order of between species. So it's not an evolution, it's just not an anti-evolution argument, it's just a different level of resolution. We'll talk about those on the last day as well. That was the setting for this. So Raup proposed the use of statistical tests to ask if this punctuated equilibrium pattern is common in the fossil record is not. So Eldridge and Gould said that species level selection might generate such a pattern, that is, some species leave more descender lineages than others, and all the variance is at that level. So Raup said, well, the logical thing is to frame these as a random walk and basically ask, is this pattern different from a random walk? What are the features of random walk? It means each step you can equally likely go up or down by the same amount, right? If you had a bias, so you tended to move in one direction, that's a departure from random walk. If you had a bias, so if you moved one way, you tend to move back the other way. So under random walk, the plus and minus changes should be equal. Under directional walk, you should see an excess of changes in one direction. Under stasis, you should see an excess of switches. You're switching too much to stay close to there. There's some simple tests you can do just based upon that. And Ralph pointed out before getting into those tests that you could have two things happening. You could have a random walk because there's no selection on the trait. Or you could have what seems to be a random walk because there's stabilizing selection on the trait, maybe even strong stabilizing selection, but that optimum itself is undergoing a random walk as the environment changes. So simply seeing the data conform to a random walk doesn't rule out selection, because you could have stabilizing selection where that optimal value is a random walk that itself is changing over time. So you'd pick up a random walk and you'd say it isn't selection, but selection could underlie. He called this a hierarchical model. So selection is driving the generational change, but the focus of selection is randomly changing, generating a random walk. So there are basically four approaches that people have used. The original was the runs test. Each step is equally likely to be increasing or decreasing. So a runs test says, are there too few runs? That is, a change in sign means you change directions. If there's a too few runs, you expect there to be half plus, half minus. If too many are plus or too many are minus, it's a non-random trend. Now, it could be an environmental trend, whatever. Likewise, if there are too many runs, it means you're jumping back and forth too frequently, right? So you overshoot, then you shoot back again. You don't expect that under neutrality, but if you have stabilizing selection, if you go one way, the Ornstein, Olan back, or whatever will pull you back. So too few or too many runs, very straightforward test to do, and you could do them. It's called a non-parametric test. There's no assumptions involved. Then there are different versions that try to get more out there. So Fred Brookstein had Brookstein's largest scaled excursions. It's related to a concept that comes out of engineering called Hirsch exponents. And then Hunt has this very nice thing we'll talk about called model weighting averages. So Brookstein's basic idea is you can put a value on the largest deviation you expect to see in your time series based upon the per generation variance in that time series. So again, using internal characteristics, instead of simply asking about plus or minus, asking about how large a deviation do you get relative to an average deviation. I think that's enough for the details on here. If you get values above this critical value, they're said to be improbably directional. That is, they're changing. They have a directionality more than you expect by chance. If you have a time series that's not very enough, it's said improbably constrained. So again, we can use directional or stabilizing selection. Again, the problem is if your environment is changing, a random appearing time series could be entirely driven by selection. You simply can't tell. Likewise, a nice directional trend could all be environmental. Amount of carbon dioxide is going up, the ocean's getting warmer, ocean's getting more acidic. You might see a trend that's entirely environmental, not genetic. But nonetheless, the ability to do tests of selection on things which have been dead for hundreds of millions of years is pretty cool. So you have to expect some limitations to be involved in that. So that's the scaled excursion idea of Bookstein. Hurst exponents is basically a very similar idea. So in a given amount of time, tau, if you look at what's called the standardized range, the initial value minus the value of tau, that thing there should scale as the square root of t. So if I do a regression, I just simply compute this for larger and larger values of tau and do a regression of this on the log of tau. The coefficient I get to the slope coefficient is called the Hurst exponent. I think Hurst, something to do with stability of dams or something. It's a really interesting idea. An h value of a half is consistent with a random walk. An h value approaching 1 is directional persistence. An h value approaching 0 is called anti-persistence, consisting with stasis. So the idea is that Bookstein's scaled range analysis and Hurst exponents are both ways of looking at run, not just looking at an individual run, but looking at the actual length of the sequence over time and asking if it's consistent with a random walk or not. So the nice features is there's nothing about heritabilities or effective population size or even time. You simply look at the time series and say what's there. Now there are obviously limitations, but the other thing is that they're nice, straightforward tests. The problem with these is they've got low power. And so there's a great expression here. Where did I see it? Yeah, basically here. Okay, so I guess it's on the next slide. Basically, it says what I said before. So here's Gene Hunt. I think he's in paleontology. I think he's curator of head paleontologist at the Smithsonian now, and he was concerned with low power for departures from symmetric random walk, created what he called the tyranny of the null hypothesis. So each of you is a fossil sequence. I'm comparing you. If the power to reject a neutral random walk is low, it means I'll include most of you as being consistent with a random walk, even though biologically it may not be true. It may simply be because I've got low power. So I have the tyranny of the null hypothesis. If I claim the null hypothesis as being directionality, I then may have a lot of directional null hypotheses. So if I change the null hypothesis, I can actually change the fraction of things that I fail to reject, right? Because null hypothesis is what you try to reject. So if your null hypothesis is directionality, you're far likely to reject it in favor of random walks. So he wanted to get around this issue. So he basically said, well, you can think of kind of three scenarios. Number one, a symmetric random walk with a mean of 0. Number two, a directional or generalized random walk where the incremental mean is different from 0, either positive or negative. And number three, stasis. And stasis, he basically assumed you're drawn from the same distribution each time. And you can then go ahead and take your data and fit each of these using maximum likelihood. So digression time. How many people have heard of maximum likelihood or ML? How many people feel comfortable with maximum likelihood? It's okay, it's fine, we're here to be helpful and friendly. So, anybody know who invented the concept of maximum likelihood? When in doubt, Fisher, right? Except Bayesian. Fisher hated what he called the method of inverse probability. So in statistics, there are three things you want to do, right? Except if you're Bayesian, then who knows what you want to do. Actually, I'm a toolbox person. Bayesian methods are a great thing to have in your toolbox, but what I hate are when you can do a t-test and someone insists you do it Bayesian. It's like, the frickin' t-test works. Don't give me this Bayesian. So anyhow, we digress. Three things you do in statistics. Number one, I've got data. I want to do estimation. What are the best parameters, right? Number two, how confident am I on those estimates? Precision. Number 3, hypothesis testing. Here's my hypothesis over here. Are these values different from those? Maximum likelihood allows you to do those all in a single setting. So the idea is, suppose each of you is a draw, and I'm assuming, never happens, that you're drawn from a normal with some unknown mean and a known variance of 1. Obviously, usually both are unknown. Let's, for now, do that. So what I would then do is, what's the density function for a normal? It's 1 over square root 2 pi, sigma squared is 1, e to the minus x minus the unknown mean mu divided by 2 sigma squared. So that's the value I would associate for a draw. So for example, if your draw is 5, then that would be the value for you. And if my draw is 7, that's the value for me. So normally the way we interpret these is we treat the distribution parameters as fixed and the data is random. If your mean is 5, how likely are you, if your mean is 3, how likely are you to get 5? Like you turns it on its head. It says the fixed things are the data. If your data is 5, how likely is the mean? Well, it means try different values of means. So basically, I have a density, so I have a assumed probability with unknown parameters, this case 1. I then multiply all my observations together. So now I get a function, in this case, that simply depends upon mu. I then simply do a plot of this function as a function of mu. I get a high point. That high point is my maximum likelihood estimator. How quickly I fall off around that high point is my precision. If I fall off very slowly, a flat likelihood, it means different values are roughly equally likely. So the curvature, and we define curvature by negative log second derivative of that. If I got a multivariate surface, the curvature is basically the eigenvalues of the Hessian matrix, the matrix of second partials. Again, how quickly you fall off in certain directions. So the high point is your estimate. How quickly you fall off is your precision. And then if I assume my MLE for mu is 6, my hypothesis is 0, I can look at the difference between my value at 6 and my value at 0, and that gives me, the difference of those ratio of likelihoods gives me a chi-square statistic for large sample sizes. So if I give you the density function I'm assuming the data come from with the unknown parameters, I can then estimate the values, their precision, and do hypothesis testing. So for example, in a QTL analysis, you've got 5 parameters. The 3 QTL means, the common variance associated with each one, and the location. So you have a 5-parameter surface to search over. And there are things like expectation, maximization, or EM methods allow you to do that very efficiently. So likelihood became very popular in the 70s. Anybody know why? What happened in the 70s? Computers. Why are Bayesian methods really popular now? Computers. MC methods are very straightforward. It's take a lot of cycles. So a lot of statistics basically tracks computational. And basically, by the way, likelihood is the engine that underlies Bayesian methods. If you have a flat prior and you assume it's proper flat prior, then the posterior of a Bayesian equals likelihood estimates. And we can talk more about that a little bit later. So that's likelihood in a nutshell, right? There's a lot more you can say about that. You can get a book on likelihood. You can read our first volume, Appendix 4. There's lots of places you can get stuff on likelihood. But it's a general tool for that. So what you can then do basically is fit each of these models with likelihood and then go a step further. So these models are what are called non-nested models. If I've got nested models, one model is a subset of the other. For example, model number 1 is unknown mean and unknown variance. Model number 2 is unknown mean. One's a subset of the other. If models aren't, and you can do likelihood ratio tests to distinguish those. If you're not subsetted, there's no formal way to compare those. People have informal metrics, things like the AIC information criteria, the Bayesian or Schwartz BIC. And basically what these do is they do a trade-off on model fitting. And the trade-off on model fitting is how good do you fit, log of likelihood, and then you penalize that. Well, if you throw enough parameters, you can make things fit really well. So the penalty is here, and this is basically for sample size, because there's n. So the corrected AIC value for a model basically says if I fit k parameters, I've got n data points, I can look at the goodness of fit, and I can compute an AIC statistic. And the bottom line is what you do if you have a series of models is you can rank those AIC statistics, and the smallest one is the best one. Now, best in what criteria and smallest in what criteria, we don't have any way to tell what AIC differences are statistically significant from each other. So it's a heuristic tool that's useful, but don't get overly excited about it. AIC and BIC are often used. They often rank models differently. The way to think about them is BIC, if I'm looking at, let's say, 20 different models, if the correct model is one of the ones I'm testing, then with probability 1 for very large sample size, BIC will pick it. AIC basically says, I have no idea what the right model is. It may not be the one I pick. Let's pick the best fitting one of these sets. So if you have to do these, report both AIC, BIC, and look at any differences between them. But what Hunt did was use this idea from Anderson. It was a nice book on model selection. He basically says, if I've got a series of models, I basically give them weights comparable to their Acquein weights. And the Acquein weights basically are, you take the smallest value, it's the best fitting, you take your value, then you use this weighting scheme, e to the minus over that. So if I've got 3 models, the weight for model 1 is its corrected AIC divided by the sum of the weights. So these, for 3 models, the weights always are positive, 0 or positive, they always sum to 1. So what that means is, you can, for each data set, say, well, given the model fit, the weights here are 0.7, 0.2, and 0.1. So now we move a little bit away from the tyranny of the null hypothesis and try to ask what you see. And when you do that, let me go to the figure here. This is a Di-Finetti triangle, and the idea here is up on this end of the 3 models, stasis, directional, or random, almost all the weight is on stasis. Almost all the weight is on random, almost all the weight is on directional. Here, for example, about half the weight is on directional, half on random, none on stasis. What you basically see is most of the weight here is either on the stasis, is in the stasis random axis. The support for directionality is fairly weak, and this is over 700 or so fossil samples. When you put together a database based upon basically marine fossils, microfossils, forams, diatoms, and macrofossils, the medium, that is the 50% value for directional selection, was 0.6, 0.47, and 0.34. The confidence interval, 95% of all the studies had a weight on directional between 4 and 8%. So very little support on here. Then you can look at, break it down by all traits, shape, size, and break it down by microfossils and macrofossils. And the bottom line here is random and stasis seem to have roughly equal support, and good evidence of directional selection seem to be largely lacking. So the nice thing about this approach is you don't have to restrict yourself to these 3 models. You can, for example, put in an Ornstein Uhlenbeck or several other models. The other thing is this is not a Bayesian approach, because what it does is, you notice what's missing is prior probabilities. It's weighting all of these models by the same amount. You may have some prior weights you want to put on these models and ask how much data change after you put the prior on. Those are called Bayes factors, for example. So the nice thing about this approach isn't that I think this is the best 3 sets. It's a good start, but you can take this approach and generalize it. And again, the purpose of this course is to kind of, review some basic ideas, but also give you a toolbox. And this AIC weighted approach, you may use that 10 years from now in a completely different setting. It's a nice, pretty general approach you can do some interesting stuff with, so just be aware of that. Yeah, sure.
Even models that have a high differences in AIC, you would still use it for averaging?
Well, remember, so what happens is here basically is that if you've got a if you have a large AIC, you get a small weight on here. Remember, small AIC is better. So that you still would, in your model, still be averaging, but let's say among those three candidates, there's very little support. So basically what the model does is it says, if you initially assume equal chance for these three models, once you run the data in, how is your support changed? And what you see is, you see increased support for symmetric and stasis, and significantly decreased perforce for directional. And again, you can try other models and, for example, have 7 or 8 different models. And in some of Hunt's later papers, he looks at some more extreme models on that. So the idea here isn't necessarily that this answers the question, but this is a nice way to think about proceeding for one class of problems, right? And that's what I'm basically showing you for a lot of these are how people have done it to give you some idea about how you may do it for your own project. Everyone's project is different. Again, don't take the problem based upon the tool you have. Use your problem to pick out what tool you're going to use, right? And this is one of many tools. So time series tests are nice, and they use just the internal features of the data to basically try to ask about something.